Today we're going to start Chapter 8, which is all about DNA. DNA is referred to as the blueprint of life. So as you listen and take notes, I want you to think about why DNA is considered the blueprint of life. All right, so to start, DNA stands for deoxyribonucleic acid. So go ahead and put that down in your notes. Okay, so make sure you know DNA is deoxyribonucleic acid. And you can see that the D is for the deoxy, the N is nucleic, and the A is acid. And hopefully you see some words in here that are familiar, such as nucleic acid, which is one of our macromolecules we learned about last semester. So if you take a look at the picture down here below, this is showing like the up close view of DNA, and then it kind of shows you how it condenses into a chromosome. So if we look at the up close view, so we see DNA, we call this picture, it's like a double helix, okay? And then in between, we have these base pairs connecting these two strands, okay? And then they're coiled, so it's that helix shape. And then this long strand of DNA, it just condenses into kind of this X-shaped structure that we call a chromosome, and then your chromosomes are found in the nucleus of your cells. In the previous slide, we looked at a diagram of DNA, and we refer to its structure as a double helix. So here's another diagram up here in the top right. Another analogy I use is just kind of think of like a ladder shape. Okay, so you have like the sides of the ladder and then the rungs of the ladder. And then instead of it being straight like my picture, imagine if you were to twist the ladder, and that's how it coils up and makes this double helix. And so the process or the journey of discovering the structure of DNA was really long and difficult. A lot of different scientists worked on this and contributed to it. Some of the most popular ones that get credit are Rosalind Franklin. So she used X-ray diffraction to create images of DNA, and she's the one that really discovered its double helix structure. So what she did, she took X-rays and she shot them at DNA, and she looked at how they bounced off to understand the shape. And then at the same time, James Watson and Francis Crick were also working on understanding the structure of DNA. And then there's a little bit of controversy here where it's thought that Watson and Crick actually stole one of Rosalind Franklin's images to understand that it was this double helix. They were the first one to get published, so these two are credited the most. So after notes today, you watch a very short video that explains how Rosalind Franklin, James Watson, and Francis Crick all came to understanding the structure of DNA. So what exactly is DNA? What purpose does it serve? So DNA is used for your genes, or think genetics. So your DNA holds the codes for your genes or your traits. Okay. So your DNA is essentially this list of instructions that tells your body how to make you, you. So DNA, we say, is your hereditary material. So in your notes, go ahead and get this whole first bullet point down. Okay, so you need to know DNA is going to code for your genes, which are traits, which is called hereditary material. And so when we talked about what DNA stands for, we pulled out the word nucleic acid. Okay. So that's the macromolecule it is. And then if you remember, monomers are those building blocks of those macromolecules. And so the monomer of a nucleic acid is a nucleotide. So go ahead and write down that the monomer of a nucleic acid is a nucleotide. And then we're going to look at, well, how do these nucleotides make up DNA as a whole? So here, the diagram on the left, it's again showing you this double helix structure. So here we have, if you think of like the ladder, this is the side of the ladder, this is the other side of the ladder, and then you can think of this as like the footsteps in the ladder. So the sides are these phosphate sugar and sugar molecules. So we call that the phosphate sugar backbone. And then if you think of like the footsteps, we call these the base pairs. Okay. And so they're all made up of nucleotides. Okay. So over here on the right side, this is a nucleotide. And I'm going to just circle one. So this right here is one individual nucleotide. We have our phosphate and our sugar, and then we have our nitrogenous base. Okay. So if we look the outsides of the ladder, we call this the phosphate sugar backbone. Okay. So if you look here, so this is the outside of the ladder, and it's just phosphate and sugars. Okay. And then the inside, where we call like the footsteps, these are our nitrogenous bases, okay, which can be A, T, C, or G. And so if we look here, these are going to be those different bases. The other side also has a nucleic acid. Okay. And so DNA is just made up of all these nucleic acids put together. All right, so in your notes, go ahead and label the nucleotide. And there's those three parts that make up the nucleotide. There's the phosphate, the sugar, and in the case of DNA, the sugar is deoxyribose. And then there's some sort of nitrogen base. And so the base options are adenine, thymine, guanine, or cytosine. Okay. And we just call them A, T, G, and C for short. And then A and G are a type of molecule we call the purines, and C and T are molecules called the pyrimidines. So in your notes, make sure you get this entire section down. Okay. So you need to know those three parts of a nucleotide: the phosphate, the sugar, and the nitrogen base. And then you also need to know the four bases that are present in DNA. So you need to know there's A, G, C, and T. So again, down below, we have some diagrams. So the first one on the bottom left, this is just a nucleotide. So here's that phosphate, here's the sugar, and then here's the nitrogenous base. In this case, this is what adenine looks like. And then again on the right, this is just showing you how the different nucleotides fit together to actually make the structure of DNA. And if you notice, what holds the bases together is a bond right here in the middle called a hydrogen bond. So hopefully in your previous graphing activity, you started to notice the trends of which base pairs like to bond together. So it turns out that all base pairs can't bond with any other base pairs. Only two can bond to each other. Okay, so what I mean by that is adenine always bonds to thymine, and guanine always bonds to cytosine. So you will never see them paired in any different order. Okay, so make sure you know Chargaff's rule, which just means adenine and thymine go together, and guanine and cytosine go together. Okay. And if you would like, you can shorten this and just write A pairs with T, G pairs with C. And so the bond that actually holds these together is a hydrogen bond. So here in the diagram, so this is A for adenine, T for thymine, and then if you see these little dots here, so that's representing the hydrogen bond that's going to actually hold those together. And then on the outside of the DNA structure, this is the phosphate sugar. Sometimes we call it the sugar phosphate backbone. And then what holds the sugars and the phosphates together is something called a phosphodiester bond. That one you don't need to write in your notes, but I do believe it's going to pop up on a worksheet later this week, so you can just come back to this slide to look for phosphodiester bond. So now we're going to use Chargaff's rule to show how these base pairs are going to go together. So this is one side of DNA, so go ahead and copy this down in your notes. And then when we write DNA, we group them in sets of 3, and I'll explain why they get grouped into sets of 3 probably next week. Okay. So let's say one side of DNA was ATA, TCA, TGC, GGG. So then the other side is going to be the complementary letter using Chargas rule. And so again, Chargas rule is A pairs with T, G pairs with C. So if this is A, the other side has to be T. Okay, we're just using Chargas rule. T would pair with A pairs with T. So then T to A, C to G, A to T. T to A, G to C, C to G, G to C, G to C, G to C. Okay. So if we want to know what DNA is going to be like, we just use Chargas rule to know what the complementary letter will be. And so why we care so much about the order of the base pairs is your combination of base pairs, so the order in which you have your adenines, your thymines, your guanines, and your cytosines, that determines what kind of traits you have. Okay. So here's a really simplified example. So let's say you have DNA that's CAT. Well, let's say that codes for purple hair. Okay. Let's say someone else has TAC. Let's say that codes for yellow hair. Okay. So if you look around the room and you look at the different traits you all have, the reason why, like, you have the hair color you have, or the eye color you have, or you're the height that you are, it's because of the combination of these base pairs that you have.